Well, welcome everyone to the Discovery and Impact series. We were very much looking forward to hosting you here on the Knight campus for this session. And we're still looking forward to it as soon as it's safe to do that. Hopefully one of the next few will be able to all gather here on the Knight campus. Our first speaker today is Paul Dalton. He's an associate professor in bioengineering here on the Knight campus, and he arrived just in March of this year. And he got his PhD in bioengineering at the Lions Eye Institute in Australia, where he worked on translating an artificial cornea to the clinic. And after that, traveled the world really through a couple of postdocs and independent career throughout Canada, Europe, Asia, and Australia, where most recently he's invented and developed a really cool technique that he's going to talk about today, which is melt electrowriting, which is a type of 3D printing. And he established that in Australia and then has further developed it in Europe, in Germany. We were really fortunate to be able to recruit Paul to the Knight campus. He's been a real pioneer in this area, published extensively on this. And I think his goals here are to think about how you can use this technique to produce new advanced biomaterials and focus on how you can get those translated into real life applications. So we're really excited to have Paul here and excited to have him talk today. And he's going to tell us more about this technique of melt electrowriting and the exciting applications for this really emerging new technology. So Paul, I'm going to turn it over to you.
Great, thank you so much, Jim. Yeah, it's actually wonderful to be here. A lot of what I'm going to show is probably new to people because this technology was originally developed in Australia, worked through Europe on this. And so now the idea is to come to the US and really see how this high resolution printing technology called melt electrowriting can be used. Today is a fun talk because I'm focusing purely from the imaging perspective. So there are going to be a lot of pretty pictures, and underlying all of that is some interesting science. So this is the title. It's a little bit long-winded, but I have a shorter one, which is not advancing. Here we are. It's called proof. And the reason it's called proof as well is that imaging for us is a proof to show that we can make a structure. The fabrics and the scaffolds that we make are very difficult to see by eye. And so if I kind of hold up this kind of fabric, it's really hard to see it. So we really need to go into the microscopic world, and there are many different methods of doing that. And just to kind of tell you a story, there was a German chemist who was wanting to be a bit condescending to me and said, "Oh, it looks like you're a professor of pretty pictures." And it's like, well, thank you. I said, you're a professor of squiggly lines. And I bring up this HNMR spectra because, this is proof to a chemist that you're being able to synthesize a particular compound. And similarly, pretty pictures are a way for us to prove, yes, we have manufactured this highly resolved structure. And so we'll go through today some of the different techniques and approaches to image these scaffolds. The technology is quite a simple one in its configuration. We have a nozzle that is run over the surface, and we have a polymer melt here. So this is can range from like 60 degrees Celsius melting point up to hundreds of degrees melting point. And we apply very, very low flow rates. So the nozzle is now much bigger than the smaller fiber. And there's an interesting electrohydrodynamic effect by applying that voltage here. It allows this thread to be stabilized. And we print at about somewhere between 5 microliters an hour to 20 microliters an hour. And so we can change the speed and we can direct write from that. This can be done on a flat surface. It can also be done on a cylindrical collector as well. And so, as you're looking at this mandrel, spinning around, we have to be exactly precise with where we put the fiber. The fibers range in size from, actually the smallest is 0.8 microns, up to hundreds of microns in size. So it's not as small as, say, electrospinning, but we can really put the fibers where we want. And in fact, we can calibrate this as well. We can change the diameter during the print. So this is an example, and this picture is purely just to show you that we can calibrate everything, and with the same nozzle in the same print, fabricate different diameter fibers to a really high degree of precision. The placement of the fibers is really extraordinary as well. We can place fibers not just kind of vertically, but we can tilt them. So on the left here, we have a fiber wall that's tilting inwards. Here we have a completely vertical, and then we expand it outwards. And so, you know, we have different mechanics in how these polymers stretch. But importantly, these are just SEM images. Video is also really important. So we created a GIF combining all of these 5. And this demonstrates that that first fiber that is laid down is in the same spot. And it also, the videos draw you in to the potential of this technique. Even though we only made 5 angles here, you can really see that we have the ability to basically tilt the fiber walls to any degree within that. And so videos are just an important medium as pictures. So when we look at these scaffolds by eye, here we have a scaffold being printed. This is mid-print. So here's the metal electrowriting head. And you can just see here, there's a faint line, and that's the fiber being laid down. So at the moment, it's printing from left to right. The scaffolds can be made quite thick. This is about 7 millimeters in thickness. And so photos, you know, you can see things, but you can't see a lot of the detail. There's also videos that we take of the printed structures as well. How we design the scaffold really impacts the mechanical properties and the dynamics. Here, this is designed to snap into location. The material itself, I'm talking about mainly is polycaprolactone, but there are some exceptions. Many polymers can be processed this way. And as an example of, you know, the different materials, we have polycaprolactone on the left, and we have polylactide caprolactone on the right. And so what's interesting is that as you have some lactide component in here, the semi-crystalline form of PCl can't exist, and so you now have an amorphous polymer. And so this allows you to stretch, and you have this recovery that just doesn't happen with the ductile polycaprolactone. So keep in mind that even though a lot of the pictures I'll be showing are polycaprolactone, PCl, this is not the only polymer by a long shot. Many polymers, in fact, I think most polymers can be processed in this way, as long as there's no thermal degradation that affects the process. Now, as we use stereomicroscopy, we can start seeing some of the details in the scaffolds that are made. We have stereomicroscopes in the lab. We basically have printers that print the structures. We take them off, and to actually see the form, to see the structure, to confirm whether we've printed what we've intended to print, we use a stereomicroscope as a matter of course. And so in this instance, we have actually, these are inverted sinusoids. Actually, this is the printing path, and they're all interlinked to create these circular structures. Or here we actually have this triangle-shaped structure, and you can't quite see this, but I'll show you later that in each direction the walls are tilted slightly by a few degrees, which changes their mechanics. So stereomicroscopy can also be used for creating videos. This is a platinum-coated tube that allows you to kind of get better reflection out of the particular structure. On the right is actually a polyoxazoline. It's inherently fluorescent due to UV light. And when we add water, this polymer will actually absorb the water and become a hydrogel in quite a rapid manner. So normally this polymer on the right is purely transparent, both in its printed form as well as in water. So we need to use things to allow us to visualize. Here we've got the UV light to help see the scaffold. in this particular example, we have a dye that we've added to the polymer post-manufacture that allows us to be able to see this being dragged up and injected out. And this aspiration that is done here can be done over and over again. There are no kind of loose fibers in here, and it is it's a water content of about 88% water. This is an interesting application as well, and also demonstrating the videography of a tubular structure. So what we have here is we have a tube with these vertical lines are melt electrowritten fibers, and there's two fibers that are also melt electrowritten that go along the length. Perhaps you can see them here. The end is plugged with a Teflon cap, and this is all filled with PDMS. And here you can apply air pressure. So now when we apply pressure into the system, it's going to stretch and expand. However, we've programmed in how it will expand. And so this is a normal speed. One of the nice things about this approach is how quickly you can get this structure to actuate. The next is an incredibly Australian experiment where actually the research engineer who's in my lab here in Oregon was in Australia at the time, and he has a burger, some burger meat here. He went outside with burger meat, and he has these 3 actuators. And the demonstration of this is to show how quickly something can actuate. This is all done in high speed. And the idea here is to be able to capture a fly and then release it without harming it. Okay, so the fly's kind of struggling and then sort of gives up. And the fly will fly off soon, but it's actually as Simon has released the pressure from the actuator. So right now you can see the pressure is being released. The fly thinks it has a chance of getting out, and it's correct. And then this actuates back to the same speed. So, you know, high-speed videography is also really important for visualizing some of the components that we make. And one of the issues that we have, and it's a major defect, is this instability. If you're not working with all the balanced parameters, you have something called fiber pulsing. And you can see this liquid-like jet coming out. And as this large amount of the jet has come out, you'll see now it's coiling. And when we print, it is really similar to, say, honey falling onto a surface. And this buckling and this coiling is due to the physical impact of the collector and the jet. So if we now use stereomicroscopy, but we use kind of a certain lighting, this is a series of fibers that are printed in this direction. And if we slow down the collector to allow some of this liquid coiling, we start off with some sinusoidal structures. We start up with these side loop structures. We end up with the figure of 8s, which if you are an expert at writing honey onto surfaces, you'll be familiar with. And then even slower, we end up with another level of coiling and buckling. So we do operate in this state between molten and solid, and that transition happens after it's landed on the collector, but very soon after. Melt electrowriting is has exceptional access to the jet and this visual component. So, you know, we can look at things like the volume of this Taylor cone and actually map that out and discover when there are instabilities or not. We can look at the jet angle, and we've correlated in this experiment the actual diameter of the fiber to the jet angle by using electron microscopy of the actual sample here. And we can see that it is it has certain diameters at different layer heights, and that it is quite constant. The infrared imaging, of course, we've used. Here we have it hot, above 60 degrees Celsius. and then you have this cooling and solidification occurring. So thermal imaging is also something that we're really interested in doing more here in Oregon. So photography allows you to get to a certain limit. What we have here are 800 nanometer diameter fibers that are stacked 50 fibers upon each other. And when that happens, you start having this kind of light refractive effects. This is a very old microscope that we have. This was many years ago now, but you can actually now start seeing the details of this. And electron microscopy is really one of the standard techniques of imaging melt electro-written scaffolds. I could probably give an entire talk just on that. But this is an example of a scaffold that is printed. This is 100 micron gap, which is actually about the lowest limit of conventional 3D printing. And so I'll just sort of zoom in here. You can see we have 2.5 micron diameter fibers spaced 50 microns apart. And there's a lot of regularity in what is being printed as well. Zoom out. And because we can change the fiber diameter, we can actually do things like create gradient structures. So at the top we have 4 micron size fibers. At the bottom we have 40 microns. Let's flip that over. And so these go down in 2 micron diameter increments. There's the embossing of the first layer of the fiber on the collector, but we have really exceptional control of whatever diameters that we would like to make. So this is a great tool for discussions across disciplines such as engineering, science, biology, because we can all understand what size features we would like to have in a printed object. In this instance, this is a scaffold that was designed by a dentist who was interested in the periodontal space and actually wanted to have a scaffold that is anchoring into muscle. So this is 10 microns, spaced 125 microns apart. Flip this over to 25 micron diameter fibers that are 250 microns, which is more conducive to bone integration. And so, a dentist can actually understand the concepts of what we're trying to do and then base their fabrication on the diameters that they'd like to make. Of course, we don't have to print straight. This is a box structure with a full sinusoid. You'll notice it's actually tilting inwards. And so what we've done in this instance is we've kind of looked at how we can look at a heart valve. And this is actually not my work. It's some colleagues of mine in Australia. So they looked at the structure and the mechanics of a heart valve, where if you make a heart valve and implant it, it has to work immediately. They basically reverse engineered the mechanics needed, and they combined this fiber with a hydrogel. And so this is now the heart valve that's in a bioreactor. And so this is meant to mimic the mechanical properties of a heart and are able to kind of achieve that. This is just one soft tissue in many soft tissues around the body that do need attention due to injuries or diseases. So this world is huge in the whole fiber reinforced hydrogel component. These materials in general have been used as scaffolds for tissue engineering. We're looking around about fibers that are about 10 microns in size, 5 to 10 microns is normal. And so when we look at these kind of scaffolds, we can send seed cells, and we want to look further into that. These are spheroids now. So this is clusters of cells that are made. Gabriella Lindberg, who's going to be another Knight campus faculty member, really specializes in cell spheroids. And so, we can place these cells on scaffolds, but like in, many instances, so black and white looks a bit boring. And so there's, ways of doing false color imaging of scanning electron microscope images. This can be done with just fibers themselves. So for example, we have 3 different diameter fibers that we print on top of each other. We'd like to really highlight the differences in the diameters. And so we can false color this and sort of show these different size fibers. You can see also how the polymer cools depending on the fiber diameter to have different sort of crystallinities as well. And I mentioned before, small changes in the printing really affect the mechanics. So this is now a false colored video of a scaffold that's made, and hopefully this works over Zoom. This all would be much, much better on the big screen in person in a seminar room. But I just wanted to show you this scaffold, this fiber here, this fiber wall is tilting inwards. And so when you pull on this from this side to this side, the top one is going to take the load first. The green level is perfectly vertical. So as you pull from here to here, they all take the load normally. And in this one, the bottom fiber takes the load first. And so this is like even more stretchy. But this is a way of conveying that the coloring and the SEM imaging is a way of showing all that. And so we can highlight different parts of scaffolds that we have and tubes that we make just using false coloring SEM. There's ways of actually looking at scaffolds from a post-processing perspective. Again, I don't know how well this works on Zoom, but it allows us to kind of take a picture that's at high resolution and then after that, zoom in and have a look at the area just to demonstrate the kind of resolutions that are achieved. Here, the fibers are about 10 microns in size, 250 micron spacing, quite standard materials for metal electrowriting. Stop motion is what I showed earlier, where we can take multiple images, combine them into one to really give an emphasis on how things could be designed. This is also stop motion done here in the Knight campus, where you're taking turns around a tube that is being made, and you take all of these different photographs, you put them together, and you create this kind of video, which gives you a visual of the actual sample. Small diameter fibers can also be suspended with this technique. So in this instance, we put very small fibers, about 2 microns in size, and we control the height of the spacing with these walls. So, at this point, we have 6 fibers that define the height of this is 80 microns, 12 fibers, 24 and 18. And this looks really nice on the electron microscope. As we now put cells on here, so this is a dorsal root ganglia that is in culture for 21 days. The neurons will project neurites out here, as well as Schwann cells migrating out. And so, this really does give a nice visual, three-dimensional, well, not really three-dimensional, but, you can see that there are fibers out of focus in here. So, this works quite nicely with the staining for, I think, S100. Actually, neurofilament. Now, as we get into the fluorescence world, this is where the technique starts struggling. I'm going to pause the video just here because you can see the fibers here, but as we play forward, we are able to see the cells, we're able to see some of the immunostaining, but we can't see the fibers anymore. These are not visible. And so, this is one of the areas where the imaging capacity of metal electrowriting really is limited. Fibers can be visualized by post-processing. DAPI actually absorbs really nicely onto the fibers, so you can see this blue staining. But it's not very consistent. It washes off with time, and also you've got to stain the nuclei. So this is really an area that we want to be improving on, and I'll come to that shortly. When we add a fluorophore to the fiber, because it's heated up to 60 degrees, the fiber actually, the fluorophore decays with time. And so, this is every 30 minutes, basically we're losing the intensity of the di-fluorophore that we add to the fiber. But it would be great to be able to visually see where the fibers are, as well as staining cells. And so, this is one of the things that I'm really interested in, and I've had discussions with the last University of Oregon Discovery Series speaker, Ramesh Jasti, about incorporating nano hoops into each of the different materials. So when we print different diameters in a multi-material way, we can actually visually see, oh, the red fiber is 5 microns, the green fiber is 20 microns. And this just gives you immediate visual clues on how the different diameters are changing, and this is something that we're looking at to the future. Now, I just kind of want to. I've shown you a lot of pictures in this period of time, and hopefully it's been interesting. There are some amazing things still to discover. And I really believe that we've only just started understanding this technology. Here is a single fiber that I've shown you many fibers printed down, but this is a blend of polymers. And so this is a blend of polycaprolactone and polyvinyl acetate that don't really combine well with each other, but we've been able to print them. When we add 70% ethanol to this, that single fiber splits into thousands of fibers that are now in the 100 nanometer size. Very, very similar in shape and structure to collagen. And so, this is fascinating. The other thing is that fiber that I showed you was with a single nozzle, but we can also do core shell. Here is an example. Oops, sorry. Here's an example of where we've pushed air into the Taylor cone, and we can actually do core shell. So this fiber is about 10, 11 microns in diameter with walls of 3 microns or 4 microns. And we have this perfectly centered structure. So doing core shell structures, and probably due to the laminar flow that we're getting, we can be even increasing the complexity of these kind of multi-component fibers. There's still so many functionalities that can be taken care of. So with that, I'm going to conclude my talk. I've really focused on imaging, lots of pretty pictures, and I believe that these are really essential, or images are essential for proving the manufacturer is there. If you're going to take pictures, you might as well take them as well as you can. It really does also allow rapid feedback for improving the process to make things in a much better way. This technique just marries so well with SEM imaging. Stereo microscopy is a way that we look at all of this, but we do have weaknesses in being able to fluorescently see these scaffolds, particularly in the presence of cells and the presence of stain cells. And also the core shell structure that I just showed is really, really hard to visualize using confocal and that because of the dimensions. And so, focused ion beam where you're cutting fibers is really the only way to prove that we've done a core shell fiber. And unfortunately, that just takes too long. So, ways in improving this is definitely something that I'm interested in. Just to finish, this wasn't done by me. This was done by a team of awesome people. I'm very lucky to have 5 people come from my lab in Europe during a pandemic to here in Oregon in March. And, we're really looking forward to talking with people, seeing where applications may be interesting for you all. And I think with that, I'll stop my talk, and I am happy to answer any questions you may have.
Thank you very much, Paul. That was a great overview. I think there's one question from John Rogers in the chat. John, do you want to ask your question directly, or do you want me to read it to Paul?
Well, I mean, I can always read it out as well. So
he's asking if you can print 2 materials with different heads to build even more complex structures by dissolving one of the materials. Also,
yeah, I've tried to build like boxes or other things, a sphere, and it can be really hard if you can't build a substructure.
Yeah, so for sure, we've printed with multi-materials. You need 2 different heads.
Yep.
However, one of the things that we really want to do is like a single nozzle that switches materials from one to the other. And there's some great work done by Jennifer Lewis at Harvard showing this can be done with regular 3D printing. And so Simon, the engineer, is really interested in having like it's basically based a little bit on microfluidics and pressure switching. But yeah, to have a single nozzle where you can switch from one material to the other and integrate some of the properties that you may want to have, whether it's not just dissolving, but maybe swelling in a different kind of way. And so there's a paper just coming out where we have different hydrogels that swell to different amounts. And if you could put this down in an intelligent way, you put it in water, it's going to buckle and fold in a way that you could have a predictable outcome.
Well, that was the other part I was thinking about, was if you applied a voltage, like you were showing the fly trap, can you have it, respond so that you apply a voltage and it moves and then releases?
Yeah. So piezoelectric polymers
Yeah,
have been
yes,
something that
right.
we've looked at, and we've been able to print PVDF, the copolymer PVDF TRFE, as well as the terpolymer PVDF TRFE, CTFE, and that is the one that is the most interesting because you can actuate this.
Yeah. Now, also, I was noticing you talk about a Taylor cone. You talk about the voltages. So these are electrospray techniques that are also used for mass spec to ionize molecules. Other strategies have been used, like ultrasonic spray. Have you used other strategies for creating that that tip? Or
I think that the
filament?
ultrasonic approach is more to create a droplet breakup, whereas I think here the focus is more on maintaining a nice continuous
Consistent
polymer fluid.
flow. Yeah. Okay. Oh, very cool. Neat. Thanks.
Thank you. The next question is also, can materials like nanoparticles or beads be suspended in the polymer and printed? Absolutely. And one of the really cool things is it doesn't really change the print much. We've put iron oxide particles into the PCL up to 30 weight percent, and it's only at 30 weight percent is some kind of effect. Have you looked at directional orientation of materials in the printed lines? Yeah, there's multiple things happening. First, if you get the fibers small enough, you can get flow-induced crystallization. So the polymer chains are kind of aligning. If you have a phase-separated material like the polycaprolactone polyvinyl acetate combination, that actually also stretches the individual phase separation. So you again get alignment there. And if you think about all the different possible polymers that are interesting to kind of add and put in there, it's a very big rabbit hole to start investigating.
Well, I have one last question for you. You highlighted some examples where, cells or spheroids were grown on these materials and were interacting with cells. Is there ways that you could maybe functionalize these materials? And so that's kind of part one. And what do you see are challenges for more cell-based applications with these materials and things we need to kind of focus on and improve on just for sustained cell-based applications?
Yeah, I mean, it's a good point. We print polycaprolactone. That's not the best cell adhesive polymer out there. And so we have done some approaches both with reactive macromers as well as something called surface-induced polymerization and photografting, which allows us to put biomolecules or hydrogel coatings onto the surface that actually either prevent cell adhesion or control that. That is definitely something that is done. But the nice thing is the chemistry has kind of already been investigated for PCL with flat sheets. Some of the big challenges of working with sort of cells and that is mainly output. Yeah, I mean, if you wanted to have 100 samples for cell culture, that might take us at the moment a few weeks to make, depending on the size as well, And so, the output is definitely something that we're addressing to just make enough. And currently our lab makes more than enough scaffolds for, say, in sort of local in vitro work. But if you want to start doing high numbers of work, it's harder.
Definitely some areas to follow up on with you for potential collaborations and discussions. I'm going to take one last question from Eric before switching over to Dan, just to give him enough time. He said, very cool images. How do you convince yourselves that what you image isn't an artifact of sample prep or the imaging experiment itself? Yeah,
sure. I mean, for things like the lower end resolution, so the lower resolutions like stereo microscopy, electron microscopy, we're kind of at the visual stage where we can actually see this in many circumstances and reiterations of the structures. The areas where it gets trickier is like core shell. And I actually didn't show this, but we were doing fluorophores both in the core as well as the shell. And then we were looking at them. The student was absolutely convinced, oh, we have core shell structures. And then we're looking further into this, and it's like, actually, no, it's the reflectance on the glass substrate coming out. It's a really good point, but this mainly manifests itself at the higher resolution levels of imaging, and you'd probably try and do it by checking different ways of visualization. So the nanofibrillar fiber that dissolves into many fibers, of course, this was checked on electron microscopy, and yes, this is exactly the case. But it's a really good point, and I think the imaging is a way of, you have to try different techniques to do it. Easy at low resolutions, much harder at high.
Thank you, Paul.
No worries, thank you.
So I'm going to introduce Dan. Dan is our second speaker for today. Dan is a staff scientist in our chemistry R&D group at Thermo Fisher. Dan has received his master's degree in chemistry from University of Colorado, studying RNA structure and function. He's been with the company for 10 years, developing reagents and imaging media for fluorescence microscopy. And he's currently leading efforts to develop products for 3D cell culture growth and imaging. And today, Dan is going to overview practical considerations in mounting media selection for optimizing fluorescence microscopy experiments. So with that, I'll switch over to you, Dan.
Yeah, thanks for the introduction, Alexia. Can you hear me and see my my PowerPoint presentation?
Yes, we can.
Awesome.
Great.
Cool. We can dive in then. So thanks everybody for hanging out for the second half today. Today's topic is mounting media, and while these aren't fluorescent reagents, which you're probably accustomed to hearing about from my colleagues, I think Paul made a really good point that, if you're going to take an image, you might as well get the best image you can, and mounting media are certainly reagents that can help you do that with your fluorophores in a biological imaging experiment. And so today, I want to tell you about, maybe I'm not meaning for this to be a history lesson, but, back probably since about 2013 or so, mounting media have been an active focus for product development and research here in Eugene. So I think what I'm hoping for today is to be educational, and really I want to share some pointers and address some common misconceptions and questions that, I've noticed over the past 8 years or so of working with these reagents. And I am not advancing here. There we go. So today, I'd like to make everybody familiar with mounting media and kind of provide some education on how to choose these. And this is particularly relevant for folks that are actively performing immunostaining in their day-to-day, but will hopefully be helpful for those that are interested. And then, might be some things to think about for folks that are developing and studying fluorescent molecules, and something that should be on the radar for those of you that are interested in image processing and data analysis, because these can have an impact on the outcome of an imaging experiment. So today we'll discuss, what are mounting media and where do they come in the workflow, and what are different types of mounting media that are commonly employed for fluorescence imaging. And then I'll walk through some data highlighting the key functions of mounting media, and then we'll wrap it up. So to help get everybody oriented here, this is a very high-level overview of the fluorescence imaging workflow. And so the focus today is going to be on fixed or dead biological samples. So we won't be covering live sample imaging, and the focus will be on fluorescence, so we won't be covering reagents compatible with necessarily histological stains. And mounting media actually come in towards the tail end of the staining workflow. So by the time we employ those, we've prepared our samples, we've applied our fluorescent reagents, whether they be immuno reagents, functional dyes, or potentially even fluorescent proteins. And really, mounting media are the last reagent that we apply to our sample prior to imaging. And while they are complex formulations and they have quite a big shoes to fill, they tend to be considered simply a glue that holds your sample between two pieces of glass. And so over the years, what I've noticed is that, a tremendous amount of focus is put on the upfront portions of the fluorescence imaging workflow. So, it could be months of use of developing a biological system to study and then, weeks to potentially months of optimizing and choosing the appropriate fluorescent stains to image biological structures of interest. But when it comes to the mounting medium, a lot of times researchers will just kind of default to whatever happens to be in the refrigerator in the laboratory that they're in at the time. And this potentially can have some negative impact, and it really can undo, months of hard work in preparing a sample for imaging. To highlight this, I want to show kind of an extreme, but an all too common example, where here I've applied a Lexfluor 67 phalloidin stain to some cells. So I'm labeling actin here. And prior to adding a mounting medium, I want to go and look and make sure that my staining was successful, and this all looks like actin, so that's great. I proceed with mounting, and I come back maybe several hours later to have a look at my cells, and after mounting, my fluorescence is all but gone. And this is a common troubleshooting discussion that we have with researchers here in Eugene. And we generally have a couple of questions when this is observed. First, what fluorophores are being used? And a lot of times we see it's like a Psi 3 or Psi 5 type dye, Lexfluor 647 included in that. And the second question is what mounting medium is being used. And a lot of times, we'll find out that it's a homebrewed recipe, or there's a couple of commercially available products out there that's from other vendors that we're looking out for. And you have to forgive us, but we do like to offer or recommend our own products where we can. So a lot of times what we'll recommend is going back and trying one of our slow fade or prolong reagents with that vial of fluorescent stain that was being used and is in question. And we'll find that actually it's not the fluorophore, the dye that's the problem. It's actually the mounting medium. So really what I'm hoping to do is pass on some information that's useful and maybe help to prevent this from happening to you in the future. All right, so the first decision that one has to make when choosing a mounting medium is whether to use a curing or a non-curing reagent. Curing mounting media require a hardening step prior to use, and this brings all the components of the mounting medium into the proper working concentrations, and it locks your sample into place. And generally, this curing step, folks will generally wait about 12 hours, but depending on the application, it can take about 72 hours or so. The curing step for the vast majority of purpose-designed mounting media is through evaporation of solvent. So there's no chemical reaction that's occurring underneath the cover slip. It's just evaporation of solvent. And in terms of solvent, there's two flavors. There's sort of the aqueous-based curing mounting medium. These are probably the most commonly used curing mounting media for fluorescence imaging. And that would include our Prolong gold, glass, and diamond anti-fade mounting media. But there are examples of organic-based mounting media out there that folks will use. It's worth being aware of. And so curing mounting media are great because they lock your sample in place and makes it quite a bit easier to handle over the long term, and it really protects the sample and affords you a little bit of flexibility in your imaging. So you don't necessarily have to image right away if you don't want to. You have a pretty decent lifetime from your sample. One thing to be aware of is depending on your sample, it can cause some compression due to the evaporation of solvent. So it's something to keep in mind. Non-curing mounting media, on the other hand, remain fluid, and in most cases, you can image immediately after application. So we offer our slow fade, gold, diamond, and glass mounting media would be examples of these from our house brand. And while you can image immediately, and that cuts down the time to imaging, they tend to be a little bit messier to work with. The slides will slide around a little bit, so you do have to use a sealant to kind of keep everything in place and tucked in nicely. But sample compression, since there is no evaporation, tends to be just less pronounced or non-existent for these. And regardless of which of these formats you choose to go with, there are options available that have nuclear dyes formulated, which offer some convenience, and I'll show you a practical application for these for archiving towards the end of the talk. And really what you're weighing here is when do I want to image? Can I wait a little bit? How long do I really want to store these images? And will I go back and look at them in the future? And then is compression something that my measurement system is sensitive to? So with that, we'll jump into some data, kind of highlighting what I consider to be the 3 core functions of a mounting medium, and that's to protect your fluorophores from photo degradation, tuning the optical properties of your sample, so refractive index matching, and then we'll touch on sample archival. So starting with protection against photo bleaching. So photo bleaching is the unfortunate loss of fluorescent signal while imaging. So we can't just turn on an excitation source, and unfortunately we can't just look at our fluorophore forever. And what this looks like when sitting at the microscope is highlighted in this top panel, where we've stained HeLa cells with a fluorescein labeled probe and applied a constant excitation signal over the course of 60 seconds. We can see as we take images at regular time intervals, there's a pretty marked decrease in our fluorescence signal. So this is photo bleaching. Before we get into where mounting media come into play, I like to call out a couple of things that can influence fluorescence retention during imaging. And sample type is one thing. So I found that fluorophores tend to photo bleach slower in tissue versus cells. And then certainly fluorescent probes can make a pretty big difference here. So in this particular example, if we use Elexfluor 48, and, for instance, instead of fluorescein, photo bleaching really isn't much of an issue over this 60-second time course. When sitting at the microscope, if we limit our exposure times and the intensity of the excitation light, that's another way to get a little bit more fluorescence out of our fluorophores. And then in addition to anti-fade compounds in a mounting medium, which I'll cover in a moment here, things such as pH and viscosity of your imaging medium, we have found can also play a pretty big role in protection against photo bleaching. So the major way that we do this is through the inclusion of anti-fade compounds, which are molecules that tend to react with oxygen. We do this because photo bleaching proceeds through destructive reactions between reactive oxygen species in your sample and your excited fluorophores while imaging. And we can see now, if we compare 2 different mounting media, one with anti-fades and one without, over the course of this 60-second time course of constant illumination, again, labeling with fluorescein, we just really don't see any noticeable decrease in our fluorescence intensity when we include some molecules that kind of help clean up the oxygen in the sample. We tend to be tight-lipped, and unfortunately, when trying to look into this a little bit more, most commercial vendors also tend to be tight-lipped about what we're actually putting into our mounting media. But if you hit the internet and start looking into mounting recipes that are commonly recommended in various circles, you'll see a handful of molecules that rise to the surface that folks will commonly use in homebrew recipes for anti-fade compounds, and these include things like N-propyl gallate or scorpic acid, Dabco, and paraphenylene diamine. We've tested a lot of these, and in our hands, no one of them has been a home run. And actually, probably what we found is that anti-fade compounds aren't necessarily one size fits all sort of deal. And so if we come back to the example where we lost fluorescence upon mounting, this is a good way to highlight this. So while we can address signal retention and we can limit photo bleaching during imaging, there are some unintended consequences of including anti-fade compounds in a mounting medium mixture that we need to be aware of. And that is that it can affect your overall fluorescence intensity and signal to background. And so in this example, my hypothesis is that this is what's actually going on here, just after years of screening anti-fade compounds and seeing what can happen. This other mounting medium here, it's probably just a incompatibility issue between Elexfluor 647 and one of the anti-fade compounds in the mounting medium. And this is something that's a bit of a problem, especially when you start to move towards multiplex imaging assays. So that's including 2 or more fluorophores in your imaging assay. And that different fluorophores respond differently to various anti-fade components. So kind of balancing your prevention of photo bleaching and keeping in mind that we still need to be able to see our fluorophores is pretty important. And to kind of highlight some of this complexity, I am showing on the left here an example of a design of experiment where we were screening different anti-fade compounds for a mounting media development program. And here we're looking at 2 different fluorophores, we'll say A and B. And for each of these fluorophores, we're interested in seeing what happens when we add more or less of these anti-fade components to the signal to background. So how bright is the sample? And what happens to our signal retention? So does it become more or less reactive to photo bleaching? Okay, and so I'm presenting these as leverage plots, and these look a little funny, but really what we're looking for here is what direction are the slopes in these plots, and where the aligned slope upwards, these clue us in that this condition is desirable for maximizing whatever our parameters that we're interested in seeing. So for fluorophore A, if we look at what happens in the presence of anti-fade 1, we'll see that while we get an increase in signal retention, so we're less sensitive to photo bleaching, we also see a decrease in our signal to background. And I pulled a couple of representative images from the sample set just to show you what this looks like. So here's the cells at a low concentration and high concentration. And at exposure match settings, we can see there's actually a pretty big decrease in our fluorescence intensity. And so this gets even more tricky once we start thinking about multiple fluorophores. So if we look at anti-fade 2 and we compare what happens with fluorophore A and fluorophore B, for instance, we'll see, well, in both cases, we do get some improvement in signal retention. And fluorophore A tends to be happier in terms of signal to background with more of this reagent in its presence. The opposite is true for fluorophore B. And I think all of this has kind of come back to the point that, mounting media are an important part of the workflow. We should probably be thinking about optimizing those, alongside of fluorescent reagents, and definitely more than just behaving as a glue in this case. This is sort of the balancing act that we kind of are walking during a product development project for a mounting medium. So really we're trying to optimize the signal to background and try to retain as much signal as possible during imaging over a large portion of fluorophores. And our prolonged diamond and solidifade diamond reagents are probably a good example of ones where we tested a lot of different fluorophores and probably have the best coverage in balancing those sorts of parameters. Right, so that's protection against photo bleaching. So next, I want to talk about refractive index matching. And in terms of product development and R&D sort of problem, this is actually probably one of my favorite projects. It's been it was pretty fun, and we came up against a lot of really interesting challenges throughout this one. And so what we're doing here by balancing the refractive index of our mounting media is we're really aiming to improve the imaging resolution and the depth that we can image into a sample. And so this is where light refraction comes into play. And refraction occurs when light travels from one medium into another, and we see a decrease in the speed and the direction of light. And this is refraction. The refractive index of a material is sort of a measure of that, and it's defined as the velocity of light and the speed of light in a vacuum, and over the velocity of light in a given medium. And sort of a real world example where we see this is here on the right hand side. So this glass of water with a straw sticking out of it. We can see that as we follow the straw into the glass of water, it's distorted. It looks to be discontinuous, and it kind of widens out in the water. And that's because light's traveling through air and refracts when it hits the glass. And then again, when it refracts again when it goes into the water. And then light that's being reflected back at our eyes kind of going back through this whole process in reverse. So it's refracted each time it goes from one medium to the next. And so our eyes are able to pick up this artifact, you know, in the macroscopic world. So it's not surprising that microscopes are also sensitive to this phenomenon. And unfortunately, there's plenty of opportunity for refractive index mismatches in the light path on a microscope. And I'm highlighting that with some of these diagrams here. So there's cartoons on the far left. I'm showing a microscope setup where I'm using an air objective, and light travels from that objective through the air and is refracted when it hits a cover glass, which has a refractive index of about 1.52. And then it refracts again when it enters the mounting medium encased biological sample. And this is representative to probably the vast majority of mounting media that are used out there. A lot of them tend to have a refractive index right around 1.47, which is close to glycerol. Right, so in turn, our fluorophores, the photons that they release are refracted as they travel back toward the objective as they travel through the various materials. Microscope companies have addressed this in some part with the inclusion of immersion oil or immersion objectives. So that provides a direct path of light to the sample, but the light is refracted when it enters a sample that's mounted in a mismatched refractive index medium. So with the Prolong and Diamond, Prolong glass and slow fade glass programs, we recently aimed to address this by matching the mounting medium to the light path. So we minimize distortions to light path all the way through. And this is important because mismatches within our imaging system, those ultimately will decrease our ability to image structures along the z-axis. And then it also results in the loss of fluorescent signal due to light scattering. At the end of the day, we're not able to image quite as deeply into a tissue sample or biological specimen. Right, and so we can quantify resolution along the axial or the z dimension. So as we image into a sample, and this is very dependent on refractive index. So axial resolution comes down to which fluorophore, the wavelength of your fluorophore, and then the refractive index, and also the numeric aperture of your objective that you're using. And I'm gonna cover this quickly here, but for those that are interested in further reading, there's a really nice Nature protocol that kind of covers this workflow that I'm presenting here. But here we've absorbed 170 nanometer microspheres to a glass cover slip and a microscope slide, and then suspended them in a curing mounting media, and then mounted the system. And we can control the focal depth or how far apart those two pieces of glass are using a spacer of some sort. And what this allows us to do then is go in and collect 3D images in our confocal of microspheres at various focal depths. And this is what it looks like on to the right here. And this allows us to compare imaging media with different refractive indices. So on the left, I'm showing prolonged glass, which is refractive index matched all the way through the system, to prolonged diamond, which has a refractive index mismatch. And what we expect to see is that close to the cover glass, we shouldn't see much of a difference in the quality of the images. And indeed, that's what we're viewing with our eyes here. But where we expect to see some decrease in resolution as we proceed deeper into the sample. So when we look at microspheres that are about 120 microns out from our objective, that's where we start to see the differences. So with prolonged diamond with the refractive index mismatch, we see that image starts to splay out a little bit and gets smeared in the Z dimension, but that's not an issue in the case where we have refractive index mismatch. And just a quick word on these images. So we don't see nice little circles, or I guess spheres, because 120 nanometers is below the theoretical resolution of our confocal microscope, which is about 500 nanometers. What we can do is, using a plugin, is we can fit an intensity profile in the Z dimension for all of these images, and we can calculate the axial resolution of these. And in this case, this is essentially the diameter of the microspheres that we're calculating from this assay. And we can see that in prolonged glass where we have a refractive index match all the way through, this line stays pretty flat and we maintain a pretty good axial resolution, but that very quickly tapers off in an instance where we have a refractive index mismatch. And really, we have measurable differences about 20 microns away from the sample. So this ultimately kind of limits our ability to image most samples to about 10 microns or so. Is probably the most common tissue section, and that certainly covers cultured cells, but kind of limits the size of, say, like a 3D culture that we can image. And what this looks like on a biological sample is actually pretty cool. So before mounting, your sample has kind of an opaque milky color to it. But if we mount in a refractive index, mounting medium with a refractive index of 1.46, we see a little bit of improvement in that opacity, but we still see the tissue due to the mismatch between the cover glass and sample. But if we match the sample all the way through to the cover glass, the tissue section takes on a completely optically transparent appearance. And what this looks like on the microscope then is also super cool, because we can see with the refractive index mismatch, we're kind of limited to being able to image maybe about halfway into this 100 micron thick tissue section. But in a mounting media that is refractive index matched to the cover glass, we can actually see all the way through the sample. And here we're just looking at nuclei, but we can start to look at biological structures a little bit more in context of something that looks closer to like a whole tissue specimen with using these kind of tools. So now, instead of just looking at tissue sections that are about 10 microns thick and taking 10 separate images, which is kind of a standard workflow, we can now just cut one tissue section and image all the way through. So this image was collected.
Then just a quick, we're at 2 p.m. We have 15 minutes reserved. Just want to give you a heads up that we're
okay. Yep, and then I can't see. There we go. All right, yeah, thanks for the heads up here. That is refractive index matching. So the last thing I'll touch on, and we'll do this really quickly, is archivability, because this is one of the more common questions that's asked: is how long can I store my samples? And the short answer is potentially up to years. So here are some samples that, here are a couple examples of this. So these are some immunostained tissue sections. The one on the left is a mouse mammary. I would have mounted and stained and mounted this back in 2017. And then the one on the right was a rat brain that I would have stained and mounted back in 2019. I imaged these earlier this year. So, it's about 4 years old on the left and 2 years old on the right. And I was able to get pretty high quality images out of both of these without having to really bend over backwards and really crank up the exposure timer gain or anything like that. So if treated nicely, you can actually get pretty decent mileage out of your samples. If it is important to you to archive, then I would recommend using mounting media that contains a fluorescent nuclear dye formulated in it. So here's an example of that. It's a prolonged diamond with DAPI was used to stain some mouse mammary tissue. And I would have done these back in 2017, so these are about 4 years old. So I mounted some mouse mammary tissue that was immunostained in prolonged glass with DAPI, and I'm comparing that down below to a sample that would have done a stepwise stain. So stained with DAPI, washed it off, and then mounted. And we can see while the immunostain stands up pretty well in both cases, we do have a pretty marked decrease in fluorescence intensity or nuclei and DAPI over the course of 4 years. So this is a when we don't include the nuclear dye in the mounting medium. So let's see here. With that, we'll move on here due to time. Just a couple of other things to think about for archivability. Keeping your samples dry and ideally just at a -20 storage condition is the best way to go. Just highlighted where you might use a nuclear, a mountain with nuclear stain to increase the lifetime of your nuclear fluorescence. Generally, we like to use curing mounting media for long-term storage. They just seem to hold up better. You can seal your slides, that's helpful. And then if you ever need to go back and re-image or if you ever need to go back and try to restain or something, most mounting media are reversible. So you just put it in PBS for an aqueous mounting medium. You can free it up in the course of probably about 12 hours or so. So that's all I have. Again, wanted to touch on some of the key functions of mounting media, and hopefully I convince you that they're more important than just glue. And with that, I'll take any questions that anybody might have here.
Thank you very much, Dan. I don't see anything in the chat, but if people want to unmute and ask Dan directly, we have 10 minutes.
I had a question about you were talking about the anti-fade and how using fluorofluor A versus fluorofluor B can have opposite effects in terms of the signal you want, or signal increasing or decreasing. And the graphs you showed, it looked like as much as one went up, the other one went down. So I'm curious, do we have a like the question I'm trying to ask is there a resource that where you've already looked at what anti-fade materials to use with which fluors when you're trying to do a multicolor assay, and do customers have access to something like that where they can be like, okay, I want to have these 5 colors in it, I should use this anti-fade because it's going to minimize any kind of problems, or yeah, I think that's the question.
Yeah, so really we try to take some of that guesswork out, and for the diamond and glass, the diamond mounting media, we tested somewhere between 17 and 20 different fluorophores, and really tried to find a balanced formulation that performs really well, both in terms of photo bleaching, but also in terms of signal intensity of your fluorophore. So and we have some recommendations as to how well the various fluorophores perform in those mounting media. And then subsequently in the prolonged and slow fade glass programs, we were not aiming to hit quite as many of the specific fluorophores necessarily, but we did want to make sure that we could hit a sufficient amount to fill out the 5 common imaging channels. So we don't necessarily, we do give some guidance onto specifically which fluorophores to use with each, but generally we try to design the reagents so that the most fluorophores possible just work nicely with the various products.
I see. So it sounds like oh, sorry, it looks like Paul has a question. I think I get it. Thank you.
Yeah, thanks. I had a question about like entire tissue clearing. So, things like the Clarity protocol that Carl Deseroth and Ali Erturk have been developing. Is there any approaches to, when you're perfusing the animal, to be kind of incorporating this kind of refractive index media in this environment?
I'm not aware of any examples where that's happened during perfusion. That would be pretty cool, and could potentially speed things up a little bit. But yeah, in most cases where something like these reagents are applied, it's generally after the animal's been perfused and the tissue sections the tissues have been removed, and generally it involves an extended period of soaking, depending on the method. Clarity being a little bit faster than some of the others.
Thank you.
All right, well, thanks everybody.
Dan. Thanks, Dan. There might be one for you, Paul, I think from Adam York. He's asking about when you're printing separate polymer materials that are not, the result in phase separation. Have you any issues with delamination between the 2 materials, and how is it overcome or prevented? And he's also asking about if you've tried printing block polymer materials only or homopolymers.
Yeah, so for the first question, it's a great question. I don't really have the full answer, except that when we print the fibers, they're really stable until you add them into ethanol. So I don't see delamination happening there. If you're having 2 fibers of different types and you printing them on top of each other, then definitely, yes, they can delaminate if the materials are not compatible. We've also printed, say, physically cross-linked polymers, And when you print one fiber on top of the other, that fusion is fantastic. So, you know, there is this kind of physical crosslinks will extend between fibers to create great fusion. So with the block copolymers and tri-block copolymers, absolutely, there's actually so much kind of macromolecular organization that's happening in this very stretched out fiber. One of the things that we've found is that you can actually create like lamella along the length as well. So the chains align. You have, with a block copolymer, you have one component that prefers to be in one zone and another in the other. And so we see this on atomic force microscopy, which is something I haven't shown here, but it's really interesting. And, you know, I'm really interested to try and look into greater detail about what happens with all the amazing permutations that we have with polymers. And the one thing I didn't mention is we also see surface, like charge surface segregation. So if you have something in a core shell that's unbound, actually it mixes and actually goes to the surface of the fiber quite a lot. So, you know, what happens when we add nanoparticles to this polymer, I don't know, but I think something interesting will happen. So we, you know, this whole world of that kind of nanoscale, microscale structure. And just to kind of also give a little bit of future direction, we are looking at printing 100 nanometer diameter fibers. And we've seen that in some instances. So we're looking at how can we do that like all the time, because then you start getting some interesting effects.
Thank you, Paul. Very nice talks from both of you. I think Dan just overviewed solutions for mainly 2D imaging and went a little bit into thick samples, but there's a lot of work and better understanding needs around 3D growth and 3D imaging. And I think there's potential for offline discussions between the work that he's leading and some of the tools you're developing for potential collaborations. Two more questions just popped up. One from Taryn, I think Billy both are for Dan. She's asking, what cover slip sealant do you recommend? And is it something we're considering developing? It seems to be a customer ask. That's the first question then. And there's another one from Eric about imaging artifacts. Did we ever decide on how much of the cellular distortion from the non-RI matched mounting media was due to RI issues and how much was physical? So we'll wrap up with those two questions.
Yeah, so Taryn's question, a lot of times I'll just use paraffin. It's pretty quick and easy. So just some melted paraffin wax to tack it down. We don't currently offer a product for that internally, as you mentioned. It is something that we've looked into, but haven't proceeded with yet. One commercially available reagent that I found that works pretty well is Biotium has one. Has a slide sealant that works really well. And I am blanking on what that is called. And then, okay, so Eric. Okay, yeah, so are we wondering? Okay, squishing. Yeah, so I have not looked into refractive index mismatch distortions versus squishing. So that's something that we haven't necessarily been able to decouple. I would say that we do see compression in certain sample types. If working with a curing mounting medium, that's refractive index matched to 1.52 versus a non-curing that's refractive. And that's also refractive index to 1.52. So we will see some difference in sample thickness in certain types, but not something that we looked at closely. Is that what you're asking? Does that answer your question?
Yep, I think so. Yeah, I mean, I know we've talked about this, right? And our collaborator was pretty convinced that a lot of times when people think their samples are squishing, it's actually refractive index problems. But I mean, it's not really easy to actually prove which one is real either way. So just wondering if you had given more thought into those samples in particular, especially like we were talking about like rat brains or something like that, where the tissue is more compressible than other tissue types.
Yeah. Yeah, and I think that there probably are instances where people think that it's actually an optical artifact that makes it look squished, but I have seen side-by-side examples where there does seem to be compression. We haven't dug into anything further than that.
Thank you very much, Dan and Paul. Tony, anything to wrap up with? Great presentations.
Those were great presentations. Thanks, everyone. Have a great rest of your week. And if there's any follow-up, you can reach out to me and Alexia, and we'll make connections.
Thank
Thanks,
you.
everyone.
Have a good afternoon. Bye.
Thank you.
Thanks for everyone for attending.